Another tool that we're going to need in genetic engineering or molecular biotechnology are DNA probes. DNA probes are small pieces of single-stranded DNA to which some type of tag has been added. The tag will allow us to find the DNA probe and thus also find where this particular DNA probe has annealed. The tag or reporter can be a radioisotope, which actually was some of the original tags that were attached to pieces of DNA when we wanted to find them. There are two kinds of isotopes that can be used in scientific research. Some are heavy isotopes, which simply mean we have atoms with additional neutrons, so they have added mass, or atoms with additional neutrons for added mass and added nuclear instability. The nuclear instability of the radioisotope means it will undergo radioisotope decay. And some of the items or subatomic materials that can be released when this unstable neutron essentially breaks down and produces essentially another proton, along with that new proton, we'll also get either alpha particles, beta particles, or gamma rays. All three of those items, alpha particles, beta particles, or gamma rays, can be detected with various types of instruments. And so we can find them. When we used original radioisotope tags on the DNA probes, we simply had to lay a piece of photographic film or X-ray film over the colonies or over the source of bacteria where we've used the DNA probe with the radioisotope tag and see where the X-ray film or the photographic film changed color or could detect where the radiation had been emitted that we couldn't see with our unaided eye, but the radiographic film could detect. Well, there are some problems using radioisotopes, not the least of which are the biological hazards. All of them, alpha and beta particles and gamma rays, have health problems. They can cause serious damage to the DNA of the laboratorian or the scientist who's working with those materials. So, for some alternative materials that were safe for tags, in more recent years, chemiluminescent tags have been added to the DNA probes so we can see them, or just simply colorimetric, so we could see the color. And on the illustration on this slide, you can see that we have agar plates with colonies at the top. The colonies are transferred by simply pressing a very thin film of plastic onto the surface of the colonies, where we're just going to pick up just a few cells. Now, originally, instead of just using plastics to pick up colonies, decades ago, scientists actually used pieces of velvet. And the little fibers of the velvet acted just like tiny little bacteriological loops or bacteriological needles just to pick up a few cells of bacteria. Those were transfers, and we would transfer the tiny little cells attached to the pieces of velvet onto other surfaces like agar plates or even nylon membranes and look for the recombinant DNA that way, again using our DNA probes. But here in this illustration, we've transferred a little bit of the colonies to a piece of plastic like a nylon membrane. Then we're going to denature the DNA on the surface of that membrane and then apply our DNA probe with our reporter. Our reporter gene will be found by simply either glowing in the dark, emitting some kind of radioactive decay emission, or changing color. Here's sort of an illustration showing you how we could use color to distinguish colonies where we can find recombinant DNA. Now, on the original colonies where we have functional genes and functional proteins, the colonies show up as blue. Where we've added extra DNA, from our recombinant DNA. And we've essentially found that the extra DNA has been inserted into the gene for the LAC-Z protein production. And it's the LAC-Z protein that helps produce the blue colonies. If we've inserted new DNA within that LAC-Z gene, then when that's transcribed and translated, we don't get we have no functional LAC-Z protein. And so we get white colonies. So simply looking at the agar plate, we can see the color of the colonies, see easily where we find colonies that will have DNA inserted into the gene, or sort of the reporter gene, that we're looking for. So it's very easy to essentially pick out the colonies we want. Here we have a more specific example of the use of DNA probes, reporter genes, and DNA electrophoresis. At the top, we have 3 specimens of DNA that are treated with the same restriction enzyme. We're going to create fragments of different mass and size, and then we're going to separate those fragments in an electric field through a gel called agarose. Agarose is simply agar that's been washed and purified. So it's a gel-like matrix that will allow us to separate the DNA fragments by mass and size. Once we've separated the fragments in the electric field or during electrophoresis, then we can add our DNA probe that has some kind of reporter, either a radioisotope, color, or chemiluminescent tag. And if it's simply a radioisotope probe or radioisotope reporter, we're going to take a piece of either X-ray film or photographic film, lay it on top of the gel, and look for the different size fragments. In the bottom picture, we have 3 samples: evidence sample 1, 2, and then the suspect's sample. Based on the pattern of sizes of different fragments, or what we call the DNA fingerprinting or DNA typing, we can see that different individuals will essentially develop or generate a different pattern of fragments. Those different fragment lengths and sizes are unique to each individual. So if we take a look at and compare the DNA fingerprint, or the size of the fragments from the victim's blood sample recovered from the crime scene that's been unidentified, and then compare that to the DNA fingerprint or the fragment distribution for the suspect's blood, we can create a statistical probability as to whether or not, or how likely it is, that that sample was derived at the crime scene from the suspect themselves. That's part of the actual application or the usefulness of today's genetic engineering in modern crime or forensics. We can do the same thing for clinical samples and all other kinds of areas of interest and investigation. DNA fingerprinting and the use of DNA probes, electrophoresis, and the generation or the creation of DNA patterns or fragments can use very small amounts of sample nowadays. We can use a single hair with the hair root, a few cells from somebody's tongue after they've licked an envelope, little fragments or pieces of skin that have been shed from their hands or their arms, any kind of body fluid, as long as it has cellular material especially. Those are all good sources of DNA that can be used in forensics, paternity testing, clinical or medical evaluation. But of course, most of us are kind of concerned or curious about how it's used in crime solving. It's been a very helpful tool. RFLP analysis can be used not only for forensics, but medicine. In fact, specific diseases like sickle cell anemia, cystic fibrosis, even paternity and grandpaternity for grandparents, RFLP has proven a very useful and valuable tool. Now, when we separate any kind of biological molecule in an electrophoresis procedure, especially DNA, the gel, agarose gel, is very sort of delicate, easily torn apart, easily destroyed, and not very stable. Well, to sort of get around that problem, a scientist by the name of Edwin Southern essentially created or developed a better technique for capturing and retaining copies of the fragments that are generated during electrophoresis, especially of DNA. Using a more stable matrix like nylon or nitrocellulose, Dr. Southern would simply press the gel to this thin piece of plastic, nylon, nitrocellulose, cellulose acetate, and then using an electric current, drive the fragments from the agarose onto the plastic. Now, the plastic, the nylon, for example, is porous. The kind of plastic that's used is something that can allow liquids to pass through, but would capture the fragments of biological molecules, like the DNA. So we can see that the DNA pattern, or the DNA fingerprint that was on the agarose, is now captured onto a more stable matrix, because the nylon and nitrocellulose doesn't easily tear, and certainly isn't as easily destroyed as the agarose would be. Now, the pattern of fragments, that unique sort of barcode that shows up, that is pretty unique, or very unique to most individuals, is called a restriction fragment length polymorphism, meaning we have different shapes of sizes of fragments for each individual. Now, with close relatives having similar, pretty closely matched patterns, but usually we think of our RIFLIP, or RFLP, or DNA fingerprint, as being fairly unique. And the likelihood, at least in a large population, of finding two individuals with exactly the same RFLP are extremely high. Very, very unlikely that we would find two people with the same pattern in the same location or geographic location, unless, of course, they're identical twins or extremely closely related. In addition to southern blotting, with the electrophoresis and transfer of DNA fragments, we also have northern and western blots. A northern blot would be electrophoresis of RNA fragments with transfer to a more stable plastic type of membrane, and western blots would be the electrophoresis of proteins, again with the blotting or transfer to a plastic matrix. In addition to northern and western blots, which are not really named for any particular person, but essentially using the name of Dr. Southern to lend an idea of using the names for the different directions on the compass to label other kinds of blotting techniques. There are also other kinds like southwestern blots, and a southwestern blot would be looking for DNA-binding proteins, and even an eastern blot. So there's been lots of advances in techniques for the isolation and separation of all kinds of biological molecules. And here's an example of a blotting compass. Notice the southern and the northern blot were developed in 1975. And in the intervening years, more and more different types of blotting techniques have been developed. Here's another illustration of using RFLP, or DNA fingerprinting, and matching the pattern or comparing the pattern, and then using a DNA probe when we're looking for a specific nucleotide sequence. Let's say we have a particular disease that we're trying to identify, or genes inside of a person that have changed, or inside of a microorganism. We can use a combination of all of these techniques that we've talked about, electrophoresis, blotting, using a DNA probe, to try and find that needle in a haystack, that single nucleotide sequence of interest. Another useful technology and technique to manipulate DNA is the polymerase chain reaction. With PCR, we're going to make multiple copies of a tiny fragment of DNA in a reasonably short period of time. We're going to need a DNA template, or the starting material. We're going to need an RNA primer. We'll need free nucleotides and a DNA polymerase, which nowadays are polymerases that are derived from thermophilic extremophiles of archaea and bacteria. Now on this slide, for whatever reason, the gamma Greek symbol is somehow misrepresenting or mistakenly being used as the degree symbol, the little zero that we usually see when someone is trying to imply or represent temperature. So anywhere that you see the gamma next to a number, it actually means degrees. Now the most common DNA polymerase that's used today are the thermophilic DNA polymerases derived from microorganisms found in very extremely hot temperatures. And one of the first was the DNA polymerase derived from Thermus aquaticus, or TAC DNA polymerase. Prior to using the DNA polymerases from thermophiles, the DNA polymerase that was used in the early forms of PCR were from E. coli. E. coli grows at mesophilic temperatures, or basically human body temperatures, or room temperature. So the DNA polymerase of E. coli cannot tolerate the extreme heating that has to be performed in the first step of polymerase chain reaction. When scientists were using just E. coli polymerase, they had to keep adding more and more DNA polymerase. By switching to the DNA polymerases of thermophiles, scientists no longer had to keep adding fresh polymerase. As long as they kept it within the temperature range for the thermophile, the polymerase would keep functioning. We take a look at the top illustration. The top of the illustration shows that we have a sequence of DNA and a region within that DNA sequence that we want to amplify or make copies of. To start, we're going to have to heat the DNA to denature it, in other words, separate the two sides of the DNA ladder. We're going to bring in proper pieces of primer. Now, they could be DNA primers, but frequently we're using RNA primers. This illustration shows DNA primers. We're going to cool a little bit to allow annealing of the primers to the sequence of DNA that we want to amplify. Then we're going to heat it again to essentially an ideal temperature for the thermophilic DNA polymerase to function, which is about 72 degrees Celsius, and allow replication to occur. We will again cool the sample enough to allow the hydrogen bonding to stabilize. And by simply going from the top to the bottom of this image, we've cycled through one cycle of polymerase chain reaction, or PCR. By putting it into an instrument called a thermal cycler, which is an instrument that essentially goes through repeated steps of heating and cooling, we can make literally thousands of copies of one single piece of DNA within a short period of time, within, let's say, a 24-hour period. And polymerase chain reaction, or PCR, has now become a valuable tool in a wide variety of disciplines. There's a fair amount of controversy surrounding the original Taq DNA polymerase derived from Thermus aquaticus, because the organism itself, Thermus aquaticus, was discovered by a microbiologist in Yellowstone National Park in 1965. Now, the DNA polymerase was isolated by scientists in a few years after that, but it wasn't until the early 1980s that a scientist working for a biotechnology corporation developed essentially the more specialized steps using the Taq DNA polymerase. The PCR had actually been developed before that. The essential steps of heating and cooling had been developed before that, but they had been using the E. coli polymerase. By simply substituting the thermophilic DNA polymerase, changed dramatically how quickly and economically, relatively speaking, PCR could be done. Now, the biotechnologists who essentially perfected PCR earned millions of dollars. Not 1 cent of royalties has gone back to Yellowstone National Park, where the original Thermus aquaticus species were first isolated. So, even though these organisms were tiny, unseen organisms growing in extremely hot temperatures, they've become quite valuable tools in biotechnology and genetic engineering today. Here we have a specific example of looking for a unique nucleotide sequence or a unique gene. We have DNA from 2 different patients. We're going to use, let's say, restriction enzymes to cut up fragments of the patient's DNA. We can then use electrophoresis with an agarose gel to separate the fragments by size and mass, transfer using a southern blot those fragments onto a more stable matrix, and then use a reporter gene to try and find a particular gene sequence of interest to perhaps diagnose an illness or give a prognosis for outcome based on treatment in the genes that are found. All sort of a combination of various techniques, including, if you'll notice on this slide, PCR. Frequently, that single gene that we're looking for is virtually impossible to find amongst all the different pieces of DNA. If we simply amplify the sequences of interest with PCR, then we can more easily find the fragments. So, using various combinations of many of the techniques that we've described in this series of slides and presentation about genetic engineering, we can do all kinds of different investigations, use all kinds of tools. It just is a matter of using your imagination and applying the right tool at the right time.